So our next speaker will be Dr. Benjamin Good. And Dr. Good, you can start sharing your slides now. Dr. Good is an assistant professor of applied physics at Stanford University and a member of the BioX Institute. Dr. Good was also recently awarded the Sloan Research Fellowship and is interested in the short-term evolutionary dynamics that emerge in rapidly evolving microbial populations like the gut microbiome. So let's give a virtual applause to Dr. Good, and yeah, you can start right away.
All right. Well, thank you, Christian. And thanks everyone near and far for tuning into my talk today. I'm really excited to be having a chance to speak here at the population genomics section. And I'd like to tell you a little bit about some of our recent work looking at eco-evolutionary feedbacks in the human gut microbiome. So I probably don't need to convince this crowd of the important role that commensal gut bacteria play in shaping our health. And as we know, one of the challenges in understanding these communities is that their structure and function emerge from the collective behavior of a large number of ecologically interacting species, often with just enormous complexity when we zoom in and look at the microscopic level, which I think is pretty well illustrated by these microscopy images by Jessica Mark Welch and her collaborators. But of course, a second key challenge, and the reason we're actually here today, is that the individual actors in this game aren't necessarily fixed and can actually evolve over time by acquiring heritable mutations in their genomes. And as we know, this process can happen extremely rapidly in microbes, with some well-documented examples in things like pathogens and laboratory experiments showing evolutionary changes sweeping through microbial populations on timescale as short as years and months and sometimes even days. And if this behavior is sufficiently widespread, you could imagine it would have important implications for efforts to understand or one day manipulate microbial communities like the gut microbiome, since it means these ecological interactions could be constantly shifting under our feet. But in contrast to the ecological dynamics that take place at the species level, which I'm sort of illustrating in colors here, we currently know relatively little about the evolutionary dynamics that take place within individual species in large microbial communities like the gut microbiome. To the extent that even many basic phenomenological questions, I would argue, still aren't really well understood. So for example, what are the relevant timescales for within host evolution? A week, a year, a whole lifetime? Do we expect these resident populations to evolve much at all, or are more complex communities just better able to respond to environmental perturbations through purely ecological means? So shifting the abundances of the species that are already there, rather than waiting around for some resident strain to acquire a new genetic mutation. If the resident populations do evolve, what do the population genetics of this process look like? What is the genetic structure of a typical resident population? How important are factors like genetic drift, horizontal gene transfer, or immigration of new strains from outside the host? And finally, how do these short-term processes within hosts relate to the longer-term evolutionary processes that shape genetic variation across hosts in the broader human population? I'd argue that without knowing the answers to these really basic questions, it's hard to know what sorts of population genetic models we should even be playing around with in the first place, or how we'd go about rationally designing and interpreting data from experiments. At this early stage, I want to try to convince you today that even some really rough order of magnitude estimates from the native human system can be really useful for helping to prioritize future theoretical or experiment work. And so to try to address these questions, our group has been developing approaches for going in and measuring evolutionary dynamics in the human gut microbiome using shotgun metagenomic sequencing of human fecal samples. And the idea is that, as Jesse just said in his last talk, in principle, these data should contain enormous information about the genetic diversity present in each one of these populations, since, to first approximation, each one of these short sequencing reads is sampling from a unique cell in the population. But of course, extracting this information is still a pretty big challenge. And that's because in general, the genetic structure of one of these resident gut populations is going to be shaped by a combination of different factors. One, different bacterial lineages are going to enter the host during an initial colonization in childhood and potentially through subsequent migration events later in life. And then once these genomes are in there, they're going to start to get shuffled around according to population genetic processes of mutation, selection, recombination, and genetic drift. And then, of course, these seeding lineages themselves are drawn from some broader global population, which is shaped by its own set of population genetic processes. And this means that the collection of bacterial genomes that we observe in a given sample today, sorry about that, are going to reflect a mixture of all of these different forces. We just got done saying we don't understand them very well yet, so it's a little bit of a catch-22. To make matters a little more challenging, we rarely are so lucky to get to observe these different microbial genomes directly, but only their shadows, so to speak, filter through these really big pools of short metagenomic sequencing leads. And so what we can measure really well in these data are the frequencies of individual single nucleotide variants. For example, by lining up these short sequencing reads against the reference genome of a given species, let's say this purple one here, and then looking for locations where we observe a mixture of two different bases at that particular site. So for example, in this case, we see there must have been a mutation between T and A at this particular spot in the purple species genome. And because the A's are present in 3 out of the 5 short sequencing reads that line up to this site, we estimate that the A's must be present at about 60% frequency within this particular species, within this particular sample, perhaps with some Poisson counting noise. And so our general approach here is to try to use theoretical models from population genetics to try to help us interpret the statistics of these noisy mutation frequencies directly. And in doing so, see if we can pull out some reasonably quantitative estimates about these different evolutionary forces without first solving this complicated metagenomic inverse problem. So for example, one of the simplest things you can do with this kind of data is scan across the genome and look for locations where you observe a big shift in frequency at that site from one sample to another. And if these are longitudinal samples from the same host, this would represent something like a host-wide sweep within the species in question. Let's say this purple one here. And so if you allow me to gloss over a lot of important technical details about how we actually go about detecting these kind of changes and distinguishing them from sequencing artifacts, which are a really big deal in these kind of data sets, I just want to give you a little bit of intuition about the kinds of things we can learn by applying this really simple approach to a large cohort of healthy human microbiomes previously sequenced by the Human Microbiome Project. So this first part is all joint work with Nandita Garud from a year or two ago, and she's actually speaking the next session. So I really encourage folks to stick around and listen to her talk as well. So as a first pass, Nandita and I focused on a cohort consisting of on the order of a few hundred hosts, a subset of whom from the HMP study were sequenced at 2 different time points, roughly 6 months apart. And so with this kind of design, in principle, it should contain some information both about short-term evolution within hosts over the 6-month sampling interval, as well as longer-term evolution across hosts in the broader human population. And given the typical coverages involved here, this allowed us to measure small numbers of genetic changes in about 40 of the most prevalent species in this particular community. So what do we see when we do this? Well, when you compare strains taken from unrelated hosts, one quickly sees that most genetic diversity in this ecosystem is relatively old, and that typical strains within most species are separated by thousands or tens of thousands of mutations. The diversity is mostly synonymous, suggesting that negative selection is playing an important role in removing deleterious variants from these populations. And then finally, the rates of homologous recombination within species are sufficiently high that the correlations between different mutations tend to decouple from each other once they're separated by more than a kb or so. So if you're interested in this later part, we've recently been developing some new theory for quantifying these patterns by looking at distributions of double mutant frequencies separated by different coordinate distances along the genome. So at this bird's eye level, these across-host patterns are actually very similar to many other species of environmental bacteria that folks have looked at in the past. Now, we can also look at the genetic changes that accumulate within hosts over this 6-month timescale, as Jesse was talking about in his talk just before. And so in this case, it's actually helpful, I think, to visualize the data by aggregating the observations across the different species and examining the distribution of the total number of genetic differences between the 2 time points. So for example, these blue points here are showing you the observed within host data, which represent about 800 different host species pairs aggregated across about 40 of the most prevalent species in this particular community. And for comparison, this red distribution shows what you get when you simply repeat this calculation, taking fecal samples from completely unrelated hosts. Now, consistent with a lot of previous work on this particular data set, these data confirm that 2 strains from the same host over time are about 100 times more closely related to each other on average than 2 strains taken from unrelated hosts, suggesting that it makes sense to generally treat these as relatively stable populations. But by plotting the data on this log scale like this, you can now see this average is a bit of a misleading quantity. And actually, the observed within host data appears to be drawn from this broad and even multimodal distribution, which suggests that it arises from a mixture of 2 different processes. So in the vast majority of cases, it's worth pointing out, we don't observe any evidence for a host-wide sweep over the 6-month time interval of the HMP study. But in a small fraction of cases, about 2% of the total, we observe resident populations that differ by tens of thousands of mutations, consistent with the typical number of genetic differences between unrelated hosts. And so we call these strain replacement events, since you can show, based on what we know about typical mutation rates and generation times in the gut, they can only be caused by the invasion of some pre-existing strain from the broader human population. so that all these mutations could have accumulated long before they actually entered the host. But in addition to these rare replacement events, we can also see there's actually a larger fraction of resident populations, about 10% of the total, with a much smaller number of nucleotide differences between the successive time points, ranging from 1 up to a few tens or so. So we call these modification events to distinguish them from the replacements over here on the right. And the idea is that these are going to represent our putative evolutionary changes, which we can just start to make out now given the enhanced resolution of our sweep detection methods here. Now, of course, you might reasonably wonder, how do we know that these are really evolutionary changes and not just strain replacement events involving really closely related strains from the broader global population? And at least at first glance, it's not clear whether it's possible, even in principle, to distinguish between these two kinds of scenarios. So actually, in this case, we found it was helpful to borrow a concept from physics and examine the statistical behavior of these genetic changes under what's known as time reversal symmetry. Now, this sounds really complicated, but the idea is actually relatively simple. The idea is that under a pure strain replacement scenario, the genetic differences between successive strains should be statistically symmetric if we just reverse the direction of time. And that's because we're essentially drawing just a set of pre-existing strains from a fixed strain pool. In contrast, we expect that local evolution would tend to single out a preferred direction of time, since most new mutations that are occurring within the host are going to be biased away from the global consensus sequence of that species. So to try to test this idea, we can imagine aggregating all the mutations associated with our putative evolutionary events, and then asking how often we observe the sweeping variant in other hosts in our large cohort. This is sometimes known as the prevalence of a mutation, and so this would be the prevalence distribution of all the sweeping mutations grouped together. Now, in this case, you can convince yourself that a reversion in direction of time here amounts to essentially flipping this prevalence distribution about its central axis. And so the pronounced asymmetry we see here in the data is actually significantly higher than you'd expect by chance if you randomly permuted the direction of time in each host independently. And this provides some further support for the idea that these are true evolutionary changes. Okay. So the main thing I want you to take away from this initial 2 time point analysis is that native populations of human gut microbiota can acquire genetic differences on human relevant timescales. And these changes arise through a mixture of events that look like external strain replacement and the evolution of resident strains. Now, having demonstrated these genetic changes can occur, the really crucial next steps are to try to understand what the population genetics of this process look like. That is, how important are factors like natural selection, genetic drift, and recombination in driving these rapid genetic changes we observe within hosts? Are these changes always driven by some environmental changes like antibiotics? Or as Jesse was saying, could they be driven just by continual adaptation to other members of these large host communities? What do these sweeps actually look like if we could zoom in and fill in the intermediate time points here? That is, what are the typical selection strengths involved? Are they always driven by selection on de novo mutations or recombination events, or by selection on pre-existing variants? And do the answers to these questions matter depending on whether we're talking about events that look like strain replacement and those that look like local evolution? It's clear that to really answer these questions, we're going to require some denser longitudinal data. And so in pursuit of this goal, we're currently spending a lot of time developing approaches for leveraging denser time series data from the gut microbiome to try to understand the kinetics of this process in greater detail. So just as an example, here's some data we just wrapped up analyzing in collaboration with the Snyder lab here at Stanford, showing the abundances of different species and the trajectories of different mutations within species from a single host who was sampled 19 different times before, during, and after a 2-week course of broad spectrum antibiotics. So just to orient you here, each one of these colored lines is showing you the population frequency trajectory of a different single nucleotide variant in 3 of the 36 species we were able to track in this particular data set. And for comparison, I've gone ahead and showed you the overall log abundance trajectory of each species above in gray for comparison. Now, this is an N=1 study, but even with data from just a single host, these higher resolution time courses were already able to tell us some interesting new things about how gut bacteria respond and recover from antibiotic perturbations at the genetic level. So consistent with what you might expect, the data confirm that ordinary oral antibiotics can drive rapid genetic changes within individual species in these larger communities, and actually at a much higher rate than we observed in healthy hosts from a few slides ago. But now that we can zoom in and look under the hood here, we can see that actually many of these genetic changes don't really appear to be consistent with the simplest extinction and subsequent recolonization pictures that we often assume. So for example, this bottom population here, Listopes feingoldii, you see that it experienced a genetic change that could be consistent with a population bottleneck during antibiotic treatment. But actually looking out more broadly, we frequently observe genetic changes occurring in species that were able to maintain relatively high abundance throughout the antibiotic treatment phase. This is the case for both of these two populations here up top. Now, at the same time, these genetic changes also don't really appear to be consistent with the simplest models of antibiotic resistance evolution, as embodied by this cartoon model of a selective sweep starting from a new mutation. You know, it's clear just sort of comparing the data by eye that the observed data look very different than this simple cartoon. Instead, because we can follow these mutations backwards in time, we actually find that many of the sweeping variants were already present at low or intermediate frequencies well before the onset of antibiotic treatment in red here. And then experience really rapid expansions during or in some cases immediately after treatment concluded. Now in this case, we can actually use the slopes of these mutation trajectories to estimate something about the typical selection pressures involved. And in this case, that the dense sampling around the antibiotic treatment phase shows that the typical fitness differences between these haplotypes are on the order of about 30% per day in this fascicle arctobacterium example, and actually greater than 100% per day in the Eubacterium elegans case. But interestingly, despite these really strong selection pressures, much higher than we typically observe in laboratory experiments, few of these variants were actually able to fix in their respective populations. And actually, by tracing the trajectories forwards in time a bit, we found that many returned back towards their baseline levels within a few months after treatment concluded. So actually, similar trends were observed across many of the other species in this particular data set. And together, I think they raise an intriguing hypothesis that there may be quite a bit of additional ecological substructure within individual species in these larger communities, which is acting to stabilize these mutation trajectories against brief environmental perturbations caused by antibiotics. Now, I think interestingly, our data here show that these effects can occur both for pairs of very distantly related strains, which differ by tens of thousands of SNPs and big differences in their gene content, as well as for pairs of strains that differ by just a handful of mutations and likely diversify either within this host or its local environment. This raises some interesting questions about how these stable ecological structures could be preserved across such a huge range in genetic distance. Okay, so I want to switch gears a little bit now. So far, I've mostly been talking about genetic changes occurring within individual species in these communities, almost independent of the fact that they're embedded together in a larger community. Of course, a really crucial question going forward is whether these evolutionary changes are actually matter at all for the broader ecological or functional structure of these host communities. In other words, do evolutionary changes within species tend to alter ecological interactions between strains, as they're observed to do in some laboratory experiments in model microorganisms? Or are these genetic changes only marginally relevant at the species level? Perhaps because in larger communities, species just tend to inhabit very separate ecological niches. You can imagine the answer to this question would be really important to know if you were trying to engineer a particular gut community to reach some ecological state. Now, a big challenge is that while some isolated examples of these kinds of feedback effects have been identified, their global impact has been a lot more difficult to quantify due to the extensive heterogeneity we expect across different species, hosts, and mutations, right? We expect the answer to vary a lot depending on the identity of the specific mutation that happens, which focal species it occurs in, and what other members of the community that species has to compete with. Now, our metagenomic approaches provide an opportunity to maybe start to quantify this pattern at a statistical level by asking whether the observed examples of within-host evolution we detect with our method are associated with bigger shifts in species composition between the same two time points, which we can measure from the exact same data. So just to give you an example of what you might expect to see here. Here's one particular host from our HMP cohort in which we detected strain replacement events in two particular species. And we also observed that several other species went extinct and a few new ones invaded over that same 6-month time interval. In contrast, here's another host from our cohort in which we didn't detect any genetic changes within species. We also observed a much more modest shift in species abundance between the same two time points. Now, these are obviously just two cherry-picked examples here, but one of our summer undergraduates, Leighton Rosenfeld, did some really great work last summer to show that when you look across our larger cohort, we find some interesting preliminary evidence that both strain replacement events and evolutionary modifications are statistically associated with bigger shifts in species composition between the same two time points. So here I'm just showing you some crude Jensen-Shannon distances in hosts in which we detected at least one evolutionary modification or strain replacement event. And you can see that while there's a ton of overlap in these distributions here, we observe a systematic trend towards slightly larger distances in hosts that experience an evolutionary modification event, and even higher ones in those that experience a strain replacement. Now, interestingly, these shifts in community structure don't appear to be solely driven by frequency increases in the focal species, as you might expect based on this little simple cartoon that I drew over here on the left. So here I'm showing you the observed fold change in all of the species in which we detected a genetic difference. And you can see that many of these species experience small, or in some cases even negative reductions in relative abundance over the same time interval in which they acquire their genetic change. This is consistent with our observations from the antibiotic time course from a few slides ago, where we saw that genetic changes weren't really easily predictable based on the species abundance trajectory alone. Instead, these ecological shifts appear to be a bit more global in nature, and that genetic turnover within species seems to be associated with more frequent extinction events in other species in the community, even when the frequency of the focal species declines. So here's just one concrete example of this sort of behavior. Here's one particular host in which we detected an evolutionary modification event in one Bacteroides stercoris population, which then declined slightly in frequency. And we also observed that two other species went extinct over the same time period, one from the same Bacteroides family, and actually another from a completely unrelated microbial family. So we're currently exploring ways to try to systematically quantify whether these extinct species are systematically more or less related to the species that acquired their genetic change. Okay, so to summarize, we're pretty excited to see some preliminary evidence linking the short-term evolution of the gut microbiome with changes in its ecological structure. Of course, the challenge here with observational data like this, it's really difficult to establish the causal direction of this effect. Right, so in addition to this first scenario here that maybe we had in mind based on the cartoon that I drew, you can imagine that similar correlations could arise even indirectly if some common environmental perturbation was able to spur both the ecological and genetic shifts simultaneously. Now, to really obtain evidence for this first scenario, one would ideally like to do something like a replay experiment using synthetic gut communities. This would necessitate a shift away from the in situ system towards an in vitro setup or at the minimum germ-free mice. Now, a different way to assess the plausibility of this first scenario, we can also try to do it theoretically by asking whether similar behaviors just naturally emerge in really simple models of evolving microbial communities where all of the ecological interactions and environmental conditions are already known to us ahead of time. So to try to address these questions, we've also been spending a lot of time developing new theory for trying to predict how evolution influences both the ecological and genealogical dynamics that occur in one of the simplest possible models of microbial community, where cells compete for different metabolites that are continuously supplied by the environment. Now, the simplest version of these models assume that the different strains take up these different metabolites at different genetically encoded rates. I'll call them R mu I, and they can acquire mutations that alter these resource uptake rates in different ways. Now, a lot of previous work has used this class of models to try to understand purely ecological processes like community assembly, particularly in scenarios involving large numbers of coexisting strains, which you can access in these models just by increasing the number of resources in the environment. Now, the key insight here has been actually that one can learn quite a bit about these large communities by considering completely random collections of strains, in which these resource uptake rates, R mu I, are drawn from some common statistical distribution. And so to try to understand the impact of evolution, we've been exploring ways to try to extend this ensemble approach to consider the very first steps that evolution takes in a randomly assembled community. So the idea is to assemble random communities with varying levels of complexity and then follow the fates of the very first step mutations that alter the resource uptake rates of one of these resident strains in different ways. And then by averaging over these two ensembles, who made it into the community and then what happened to them, the goal is to drive something like a scaling relation in order to quantify how the typical fitness benefits or ecological impacts of these first step mutations depend on the taxonomic diversity or the metabolic overlap of the surrounding community. And so in this way, we're really excited about these resource competition models as maybe providing a mechanistic approach for helping us address some key verbal models about how evolution operates in really large microbial communities. So for example, should we naturally expect evolution to be slower in larger communities because there's just fewer open niches left for mutations to exploit? Or do larger communities just naturally create more ways for strains to adapt to the idiosyncrasies of their local competitors? When do we expect successful mutations to tend to replace versus coexist with their parent strains? And when do we expect them to drive extinction events in other species? Now, the challenge, of course, is that there's many different ways of modeling mutations here. But interestingly, some preliminary computer simulations using a really simple model of knockout mutations and a global energy budget already reveal some interesting counterintuitive behavior, which at least qualitatively resembles some of the behavior we were observing in the microbiome data from a few slides ago. So here I'm just showing you two different realizations of this model, showing the successful first step mutations that occurred in communities with 50 and 15 different coexisting species, respectively. And so the basic idea here is that by eliminating a pathway that the strain doesn't use, it's able to reallocate some of its limited resources to importing other metabolites that it eats. And you can imagine this could provide a fitness benefit in the right conditions and allow that knockout mutation to take over. And so what these examples show us is that it's actually relatively easy in these models to find sets of parameters in which a relatively large fraction of all successful knockout strains can manage to coexist with their parent, similar to what we observed in the antibiotic time course from a few slides ago. Now, it's maybe not so surprising that this happens in communities like this top example here, where with 15 strains and 50 resources, you can imagine there's a lot of niches left for these mutations to exploit. But interestingly, we find that this ongoing diversification continues to occur even in scenarios like this bottom one, where all the niches are already saturated by other coexisting strains. And moreover, we also find that many of these successful mutations are associated with small, or again, even negative changes in abundance of the focal species they occur in, similar to what Leighton was observing in her observational data from a few slides ago. And so we're currently working on some analytical calculations to try to see if we can understand the mechanisms behind these counterintuitive behaviors here, which we think should be possible now given the simplicity of the underlying model we've ended up with here. Okay. So to summarize, I've shown you some data today to convince you that native populations of human gut bacteria can evolve within healthy human hosts on clinically relevant timescales through a mixture of external strain replacement and the evolution of resident strains. I've also shown you some data to try to convince you that the ecological and evolutionary processes in these communities might be inherently intertwined. On one hand, we found that the genetic responses to antibiotics suggest that there could be quite a lot of additional ecological structure within species in these larger communities. Which immediately raises some questions about why these niches aren't already filled by dedicated species, which don't really seem to be in short supply in this particular ecosystem. We also found that some preliminary evidence that genetic turnover within species is correlated with global shifts in species level composition between the same two time points, although not necessarily in the abundance of the focal species itself. And finally, we saw that some of these qualitative behaviors can be recapitulated in really simple resource competition models evolving in the high diversity limit, which maybe creates some exciting opportunities to try to use theory to help us understand the mechanisms behind the seemingly counterintuitive behaviors in certain cases. All right, so with that, I'd just like to thank all of my wonderful collaborators on these studies, particularly Nandita Garud, my co-author on the HMP study I talked about in the first half, Morteza Rudgar, who generated all the beautiful data for the antibiotic time course in the middle, and Leighton Rosenfeld, who spearheaded the correlation analysis at the end. And I thank you all for listening and happy to take your questions.